everyone, my name is Smita Jandir.
My name is Isabel Robbins, and today we will be presenting to you exploring the loss of function of DNA repair proteins in the developing central nervous system.
And we worked under Professor Isabel Oviedo at the City College of New York.
So let's start with the basics of what is DNA. DNA is known as the building blocks of life, and the first thing to remember in the context of our presentation is that it is composed of 5 nucleotides. You might have heard of 4 of them: A, T, C, G, adenosine, thymine, cytosine, and guanine, and the 5th one, uracil, is in the process of RNA, but altogether these 5 make up DNA. And the second most important thing to remember is that DNA is constantly being damaged and repaired in your body, and if these damages are not repaired correctly, it can lead to diseases.
So let's look more into these different types of damages. The first is the DNA mutations. Mutations go in and directly change the very genetic coding of your DNA, and thus they're very severe and cannot be repaired. We also have DNA breakages, of which there are approximately a quintillion that your body repairs every single day. So what are these repair systems to fix these breakages? So one of these repair systems is called the DNA mismatch repair, or MMR system. As you can see in figure 4 here, there's one nucleotide that's mispaired and missed during the proofreading process. Through a process called recombination, we are able to switch out this nucleotide with another nucleotide that actually fits from an intact piece of DNA, just switching these bases in order to fix the breakage. And this recombination process is motivated by the MMR proteins, the mismatch repair proteins, MSH2, MSH3, and MSH6. So MSH3 and MSH6 bind to MSH2 in order to create sort of a super complex of a DNA breakage protein. And we are going to be focusing in on MSH2 today.
So what do we know already about the MSH2 gene? The MSH2 gene has been studied thoroughly in the immune system with something called antibody diversity. Antibody diversity is the process that your body does to make more antibodies to fight off the ever-growing number of antigens around us. So antigens are viruses, bacteria, infections that could give you diseases, and antibodies fight them off. So MSH2 participates in this DNA recombination, antibody DNA recombination process. But we do not know what MSH2 gene does in the brain. A recent study showed MSH2 and the link to Huntington's disease, which is a neurodegenerative disease characterized by loss of sensory function and motor function over time. Huntington's disease is caused by something called a CAG repeat. A CAG is a trinucleotide, cytosine, adenosine, and guanine, and if you have more than 36 of these repeats, your gene is mutated and you will develop Huntington's. If you're between 10 and 35 repeats, your gene is in the normal range and you will not develop it. So a recent study showed that in mice with no MSH2 gene present, these CAG repeats were expanded. So that means that when you had no MSH2 gene, your chance at getting Huntington's disease was a lot higher. So it's really important for us to look at MSH2 and what it does in the brain, because we can figure more about these neurodegenerative diseases, not just Huntington's.
So keeping these neurological implications of MSH2 in mind, we can say that if left unchecked 30 years from now, more than 12 million Americans will be suffering from some sort of neurodegenerative disease.
So just to sum up what this MSH2 immune system protein is doing in the brain, this is uncharted territory that we're entering. So our research is exploratory, meaning that we do not have a lot to compare it to before. And the reason that this is so surprising is because the brain has been known to be separate from the immune system. It has its own defense mechanisms, its own housekeeping genes that fight off diseases. But we're seeing overlap. So that's really what we're going to be looking into today.
So now let's delve into our experiment and what exactly we researched this summer.
Our research question was, what is the impact of MSH2 loss of function in the central nervous system? To answer this question, we had 2 mice, a knockout and a wild type. A wild type is essentially our control group, so it has MSH2, and the knockout does not have MSH2. These 2 mice were brothers, born from the same womb, so the only thing that is different between them is the varied gene.
So we used 2 different brain markers in our experiment to study the loss of function in MSH2. So the first, as you can see here in figure 7, is the parvalbumin or PV protein. We use the PV protein to determine we use it as a marker to determine inhibitory neuron density in the brain, and we also used it as a cell type specific density marker. So essentially, when we were comparing our knockout versus wild-type mice, we were comparing how many cells there were exactly in each part of the brain that we tested on.
The second marker we used was CFOS. The CFOS is an immediate early gene, but the protein that it produces is used as a marker for recent neural activity. So when looking at our images, we want to see which one expresses more CFOS, meaning has more neural activity.
So here's a brief overview of our experimental design. So these two brain markers we just discussed are also used to determine two different neural processes in the brain. So we were able to detect these markers by using immunohistochemical techniques in both the MSH2 wild-type and MSH2 knockout mice. Then we were able to image them using a confocal microscope, and from that we were able to obtain a result on neuronal activity using CFOS and inhibitory neuron density using PV.
Here's our methodology more in depth, divided into 3 different sections. The first one of preparing the brain, the second of immunohistochemistry, our staining process, and the third of image analysis and how we got our results. Going into the first one of the perfusion of the mouse in order to obtain the brain, on the top picture you can see how we lifted up the sternum of the mouse to reveal the heart. And then in the second picture we used a peristaltic pump. So we pump a buffer through the mouse, which is to clear all the blood out of its body, because during imaging, blood causes a lot of background. And then we go in with a fixative. In this case, we use paraformaldehyde, which essentially freezes the brain in time right before the death of the animal to preserve neural activity.
Next, we were able to slice the brain. So the mechanism we see here is the Pelco Easy Slicer machine. We turn the dial on it 360 degrees in order to get our slices to be 800 microns thick. Then, when the slices floated off into that little well of PBS you see there, we were able to pick it up using the tip of a paintbrush and place it in another well of PBS in order to further preserve it.
Next is our immunostaining process. So first we stain with a primary antibody, and then we stain with a secondary antibody. So the primary antibody is seen in the yellow, and it attaches to the epitope, which is essentially like a helping hand that the CFOS has to easily bind. And then we go with the secondary antibody, which contains a fluorophore. So the fluorophore is what gives CFOS expression and PV expression fluorescence in our confocal images. So when we're looking at our images, we're looking for brighter spots in the brain to show where more expression is of PV or CFOS. So in this animation, there was more expression in the auditory cortex, so that is the green right there.
So after our immunohistochemical techniques, we were able to mount our brain slices, as you can see in figure 13. We then took one of these brain slices and placed them under the confocal microscope, which used lasers to image them.
And here are the 3 parts of the brain that we compared in the knockout and the wild-type mice. So we compared the thalamus, the hippocampus, and the cortex. You may have heard of the cortex being your decision-making part of the brain, and your thalamus is for involuntary actions such as sleep and consciousness, and your hippocampus is where your memory and emotions are stored and developed.
So here's a brief example of what our image analysis process looked like. So we're just going to use the CFOS wild-type and knockout cortex as an example. So these are the raw, unedited images that we put into ImageJ. And next, we applied some filters on them in order to subtract the background and get the cells as visible as they possibly could be. Then we used the threshold tool in order to get them against a black and white background. The picture you see here is us using the analyze particles tool just to count these cells off. And ImageJ also generated a table of the total amount of cells that were present in each of the images, as well as the area of each of them. We were then able to calculate the density from this and plot them on a graph using MATLAB. So here's a brief overview of our results.
Starting with our CFOS results, we found that CFOS levels are significantly lower in the MSH2 knockout, meaning that when no MSH2 gene was present, we saw less neural activity. Looking at our graph, you can see in the cortex, thalamus, and hippocampus, very different regions of the brain. The wild type always had more CFOS expression, meaning more neural activity. When analyzing large data sets, you're looking for a p-value of less than 0.05 to determine your data statistically significant using a 2-sample t-test. So our value was 0.0125, deeming our data statistically significant.
So next we examined our inhibitory neuron marker PV. So initially we had used the same processes for PV that we had used for CFOS. However, this resulted in insignificant results. So we looked at the larger context of the brain, and with the human eye, we were able to see that there was a higher concentration of PV-expressing cells in the knockout reticular formation part of the brain. So what we did was, rather than use the analyze particles feature on image J, we used the manual cell counter, and by hand we were able to determine that there were 110 cells in the knockout and 79 cells in the wild type. So on MATLAB, we generated a histogram of the mean intensity of these cells, which is just a fancier way of saying the mean brightness. So as you can see, there's a lot of overlap in between the wild type and knockout bars here. And the p-value that we calculated on MATLAB was 0.2, still a lot higher than 0.05. So this still rules our data insignificant and our results insignificant. However, since in all the slices we had imaged, there's a higher trend of PV being expressed in the knockout, we can say that this is a result.
So here are our speculative conclusions. Due to our PV data being statistically insignificant, these are just things that we can speculate. So with our CFOS data, we saw that there was less CFOS expression in the knockout, meaning when no MSH2 gene was present, there was less neural activity. And in PV, we saw greater inhibitory activity in the knockout reticular formation. So that means no MSH2 gene, there was more suppression. So how can we link these two results together? The reticular formation's main function is to control cortical activity, meaning that if there's suppression in the reticular formation, there is going to be less neural activity in the cortex, which is exactly what we saw in our results. So this is still speculative due to the insignificant data, but it does give us a foundation and a baseline to look into for further research.
So what are some next steps we can take for further research? So one thing we can look at is how progenitors, which are essentially just stem cells of inhibitory neurons, differ from excitatory neurons. So with PV, we were able to examine the suppression of inhibitory neurons, but we can use a different marker in future research to determine how this has the same implications with excitatory neurons. We also now know where to look for different development in the brain. With our CFOS results, we were able to determine that there was development in the hippocampus, cortex, and thalamus, but we can further examine these in the future. And finally, we can use an MSH2 reporter mouse, which essentially has fluorophores injected directly into the genome to look at how MSH2 expression changes during the development of the brain.
And to link this all back to neurodegenerative diseases, the more that we know about MSH2 on a zoomed-in level is going to help us figure out the link of MSH2 and neurodegenerative diseases more in depth. So thank you for listening. We would like to thank NYU GSTEM, Dr. Oviedo, and the Oviedo Lab at City College of New York for giving us this wonderful research opportunity.
We'd also like to thank our tutor, Neelam Pandya, our junior tutor, Kara Kirchian, and Ms. Catherine Tissot and Monica Parham, and the NYU Cron Institute of Mathematical Sciences for putting this program on. Thank you.